Our moderator today is Bonita Shobe, President of the Brain and Behavior Research Foundation. Bonita, please go ahead.
Thank you. Welcome to our final webinar of 2011, sponsored by the donors and friends of the Brain and Behavior Research Foundation. I'm pleased to report that since inception of this inaugural series, just this past March, we've reached well over 1,000 of our friends through this powerful format. And the feedback from so many of you is to keep them coming, and in that spirit, we are working toward a 2012 series of topics and speakers. So thank you so much for your support. In fulfilling our mission to the 1 in 4 people diagnosed with a mental illness, the Brain and Behavior Research Foundation's Meet the Scientist webinar program is yet another example of how we realize our commitment to educating and informing our audience. As always, these webinars are free of charge and are available to the larger community on our website. I invite you to listen to archived presentations via our enhanced website. I'd also like to take this opportunity to thank all of you who have been supporters to the Brain and Behavior Research Foundation, and certainly entreat you to continue to do so. We appreciate your support. I'm now pleased to introduce a good friend of the Foundation, a Scientific Council member, Dr. William Bunney, as today's presenter. Dr. Bunney has some wonderful opening comments as well as a wonderful presentation, so I'd like to turn it over to him immediately. And thank you, Dr. Bunney, for being such a good friend and leader in our community. And thank you for joining us today. Dr. Bunney.
Bonita, thank you for the wonderful introduction. Bonita has asked me to comment briefly on my career. I attended Oberlin College, University of Pennsylvania Medical School, and completed my residency in psychiatry at Yale. I then assumed a number of positions at the National Institute of Mental Health in their in-house intramural research program, where I directed research on schizophrenia and mood disorders. During my time at the National Institute, I also spent 3 years heading up the Department of Health and Human Services Education, Treatment, and Research programs in drug abuse for the nation. Later moved to University of California, Irvine, where I served in positions as chair of the department and later as senior associate dean for research for the medical school. Although throughout my career, I have continually conducted research on the treatment, diagnosis, and search for causes of schizophrenia and bipolar disorder and major depressive disorder. As many of you know, the current medications are used for the treatment of mood disorders require 2 to 10 weeks for full clinical effect, and only a percentage of patients actually respond the first and even the second time on medication. I will share with you today research concerning rapid-acting antidepressant strategies, which are fully effective in 50 to 60% of the patients within a 24-hour period. I will also discuss what we know about how these treatments work in the body at the molecular level, since this knowledge will help us develop new and even more effective rapid treatments for these disorders. I would like to note that I have served on the Scientific Advisory Board, as Bonita said, of the Brain and Behavior Research Foundation, formerly known as NARSAD, since its inception, and consider it to be one of the most important organizations in the world in raising millions of dollars to support the research of young investigators. These monies have allowed young researchers to become involved in studies of these disorders and to develop a passion to try to discover fundamental knowledge for the treatment of these illnesses. So let me now turn to my presentation of rapid-acting antidepressant strategies. So there is an urgent therapeutic need. Long-standing challenge in psychiatry is the development of rapid-acting antidepressant strategies for mood disorders that are effective within 24 hours. Current antidepressants, as I just mentioned, take 2 to 8 weeks, sometimes 13 weeks, for full clinical response. During this time, the patients continue to suffer from their symptoms. It's been reported they're at significant risk for suicide, as well as experience harm to their personal and professional lives. Rapid-acting antidepressants make a major health care implication. Now, this is an outline of the issues that I'm going to discuss. Sleep deprivation therapy. These are all rapid. Deep brain stimulation treatment. C, low-dose intravenous ketamine. And this will be a nice continuation of research presented by Dr. John Crystal in the previous Brain and Behavior Research Foundation webinar, and then future studies. Now I'm going to start with sleep deprivation therapy. I'm going to talk about potential brain imaging biomarkers which might predict and could predict who will respond to sleep deprivation therapy. I'll also talk about circadian abnormalities which have been observed repeatedly and consistently in mood disorders and treatment implications in mood disorders. Discuss that all circadian rhythms, these are the 24-hour rhythms of the body, are regulated by genes called clock genes, potentially clock gene biomarkers to predict treatment-resistant depression. And a recent study that we published, showing that rapid, robust, and sustainable clinical improvement to combined chronotherapeutics, and I'll describe that, treatments. Okay, now there have been a number of meta-analyses. This one included over 12,000 patients in terms of the rate of response to these compounds. And on the left in white, we see the current antidepressant medications, which in these analyses combined take about 5 to 6 weeks for full efficacy. You do have statistically significant change at week 1, as shown in the bottom, but you can see that sleep deprivation and ketamine is even faster. They produce a response within 24 hours, which is significantly different. Now, that's in 40 to 60% of the patients. They produce this response much more rapidly than the current antidepressant treatments. And you can see the weeks at the bottom. All right, now, this is a complex slide, but I wanted to make the point that the current treatments, tricyclics, SSRIs, which a lot of people use, MAOIs, take 2 to 13 weeks. I said 8 weeks before, but some of them take longer in the elderly. But then going to the this column here, you can see that there's an extremely low response rate, if any, to these drugs. And ECT, although more rapid, also has a low response rate. In contrast, the 3 in yellow, sleep deprivation, low-dose ketamine, and I will comment on deep brain stimulation, which is somewhat different, have a 50%, 70%, and 60% response rate within 24 hours in a subset of the patients. And I'll discuss the mechanisms of action later. Okay. Now, let's start with sleep deprivation. It involves maintaining the patient awake for 36 hours continuously. This sounds counterintuitive, but over 75 papers with 1,700 patients studied during the last 4 decades have documented that one night of total sleep deprivation can reverse depressive symptoms within 24 hours, 40 to 60% of severely depressed patients. However, with sleep deprivation therapy alone, relapse often occurs after the next night of sleep. Okay, now this is just a patient that we studied. 47-year-old, major depressive disorder male, continuously depressed for 30 days, responded within 24 hours to one night of sleep deprivation and relapsed following the next night of sleep. His symptoms changed from psychotic, suicidal depression to mild euphoria and talkativeness following sleep deprivation. And the challenge here is to prolong this improvement or to block that relapse. And we have some data which I will present which successfully accomplishes that. This is also a remarkable presented research paradigm strategy, because the day before, when the patient is psychotic and depressed, you can collect brain imaging data, you can collect blood data, and then the day after, you can collect it. When 50% have shown a response and 50% have not, and you can contrast that data as I will show. Okay, potential brain imaging biomarkers. Let's start with that to predict response to sleep deprivation therapy. Okay, the method here was all subjects were sleep deprived for 36 hours. We happened to have 36 MDD patients. 12 responded, 24 did not. There were 26 controls, and they got PET scans the day before and day after, and they were rated on a standard rating scale for depression called the Hamilton scale. Okay, now just a note on positron emission tomography. I know many of you know what it is, but it measures regional brain metabolic activity by evaluating glucose, which supplies energy to the brain. So again, we collected PET scans the day before and then the day following sleep deprivation. All right, now I'm just going to focus on the first two figures here. This one, these two. Yeah, and this is all baseline, and this is effects of sleep deprivation. So in this first figure, you have a contrast between responders and non-responders. And the index of glucose utilization is over here. It's red is high, and green and blue is low. And you can see increased activity here in those that responded. And following sleep deprivation, you can see that it goes from the high range down into the low range, just there. Okay. And you can see no change in the non-responders and in the controls, again, this area here, in the frontal area. Okay. So in summary, increased metabolic glucose rate in the ventral anterior cingulate and the medial prefrontal cortex, which are the areas that lit up in red, could predict patients that respond to sleep deprivation therapy versus those that do not. Now, this is not an isolated finding. There are 12 studies, increased pre-treatment, baseline, cingulate, that's that frontal area, that's part of the frontal area, activity, predicts response to sleep deprivation in 12 brain imaging studies. There are 4 PET studies, 5 single proton emission computed tomography, and 3 functional fMRI studies. all consistent with this increased activity in this area. Now I'm going to change and talk for a moment about circadian rhythms, and you'll see how they fit into the whole story. It's striking that virtually all body processes systematically function during the 24-hour period, including sleep, temperature, and hormones. Now I'm going to talk about circadian rhythm abnormalities and go through some of these mood disorders. The fact that the regulation of circadian 24-hour rhythms are all controlled by genes called clock genes, and that circadian treatment manipulations in mood disorders can be effective. So now this is evidence of abnormal circadian rhythms in a subgroup of mood disorder patients. First of all, marked circadian variations in clinical depressive symptoms. Patients can be severely symptomatic during early hours of the morning with marked improvement late afternoon. Now, I'll give you a couple of examples. Abnormal temperature curve. Again, these are 24-hour curves. Increase in nocturnal core body temperature and flattening of the diurnal curve. Elevations in stress hormone cortisol with absence of late day trough. And then sleep abnormalities, abnormal sleep patterns, including shortened REM, that's dreaming, measure of dreaming, great onset, and early morning awakening, which could reflect abnormalities in the rest activity cycle. So here's just an example of this is sleep period, and you can see that depressed patients have higher core body temperature than the controls. And this is a composite graph of over 150 patients, over 1,000 cortisol assays. And you can see, this was put together a number of years ago by Will Carpenter and myself. You can see that the circadian rhythm of cortisol is elevated above controls. That's the blue line above the controls at every point. But what's striking is 2 points, and that is we all have increase in cortisol in the early morning that drops off during the day. The depressed patients have a shift in this increase for about 4 hours earlier, and they have this peak in the late evening, which the controls do not have. And then a couple of examples of stable and marked diurnal mood changes in subgroups of depressed patients. And these patients were rated every hour. And you can see here, this is severe and this is lack of depression. You can see that the yellow line represents a group of quite stable depressed patients, severely depressed. While the blue-green line shows that these patients are at about 6 or 7 in the morning, psychotically depressed. I mean, this just is a depression rating, but they're extremely depressed. And that this almost remits by 10 or 11 o'clock in the evening. Now, just an example of 2 patients. Here, fortunately, we have the reverse. So severe is at the bottom and less depressed at the top. But this patient was consistently depressed over 60 days, while the patient at the top, the green lines, fluctuated markedly, daily, with a couple of exceptions here, daily between severe depression in the morning and almost euthymia or wellness in the late afternoon. So, implications of molecular clock genes for circadian abnormalities and mood disorders. Are these circadian abnormalities central components of mood disorder or simply epiphenomena? And one test is if one manipulates circadian 24-hour rhythms, will depressive symptom pathology show profound improvement? In fact, it does. If so, it would argue that abnormal circadian rhythms, which, as I said, are completely under the control of clock genes, may be etiologically linked to mood disorders. So circadian manipulations, as I said, can produce striking and often rapid treatment response in a subgroup of mood disorder patients. Circadian manipulations include non-invasive treatments. I've talked about sleep deprivation. I will now discuss bright light therapy and sleep phase advance, all circadian manipulations. Thus, the identification of abnormalities in clock genes, which control, as I said, circadian rhythms, could lead to new knowledge concerning one of the core pathophysiological elements. Core pathophysiological elements in mood disorders. All right, clock genes. It is of interest that one of the most pervasive epigenetic environmental influences in the entire evolutionary process, from single cells hundreds of thousands and millions of years ago to man, has been the 24-hour light-dark cycle. So this is a very, very simple graph of what happens with clock genes. Positive gene activators activate, and I'm showing plus here, turn on clock genes that make messenger RNA, which makes the protein, which moves out, this is a cell now, which moves out of the nucleus, the center of the cell, out of the nucleus into what's called the cytoplasm and makes proteins. And these proteins then move back into the nucleus and shut off their own genes over, and this process takes 24 hours. Now, it is so much more complicated. I'm not even going to show you the slide. There are probably 100 genes that are involved in this entire process. Okay, potential genetic markers for response to sleep deprivation therapy, clock genes. I've talked about brain imaging. Again, we collected blood for lymphocytes just before and after sleep deprivation. For microarray profiling, which is a technology to actually study all the genes in the human body instead of the single ones we were able to study in the past, and qPCR, quantitative analysis kind of thing, which can confirm the findings of microarrays. Okay. Now these are just 3 genes that came up. Responders, and that's in blue, showed gene expression for 3 significant genes after sleep deprivation. This is pre-sleep deprivation. This is post-sleep deprivation. And non-responders in red showed decreased gene expression. This was a small study. This was headed by Mark Votter. By the way, the imaging study, which I showed, was headed by Joe Wu. All right. Now, non-pharmacological manipulations. of circadian rhythms. I'm just going to discuss this. I said I'd talk a little bit about sleep phase advance. This is simply shifting the time of onset of sleep. So the patients are put to bed 6 hours, 4 hours, and then 2 hours earlier. And bright light therapy has been used in seasonal affective disorders and in non-seasonal affective disorders. Bright light treatment usually involves exposure to bright light. 10,000 lux, a measure of light, over a period of 1 to 2 hours. So there have been 12 randomized studies, or 11 randomized studies, many of them confirmatory, but some not confirmatory of this finding. Now, this is a study, again, headed by Joe Wu from our research group, and the black line indicates the day of sleep deprivation. And these patients, because they were bipolar, received lithium 7 days before, and then an SSRI 3 days before. It takes, as I mentioned, 2 to 8 weeks to get a response. So this is not I'm going to show you, is not a response to that. Then we were able to, probably with a combination of these medications, but specifically with sleep phase advance and bright light therapy, for the next 3 days, we were able to sustain the effect. So the dots indicate the depression Hamilton ratings that were done for the first 6 days before, and then the first 6 days after sleep deprivation, and then each week after that. And here we see the results. The blue line is treatment as usual, whatever they were on. The red line is the group that received the sleep deprivation and received the sleep phase advance and bright light therapy. And the little dots above that indicate that at every point except day 6, these patients were significantly better than the patients that had treatment as usual. And you can see a rapid response with deprivation. And what was interesting is that at day 7, 50% were not just in remission, a response, but 50% were in remission from the illness. We published this paper a couple of years ago in Biological Psychiatry. Okay, now I'm not going to go through the details of this slide, but this was a review that my wife and I published a couple of months ago. And these are major depressive disorder patients. And a number of people have done follow-up studies greater than 2 weeks using sleep deprivation. And 5 of these 6 studies reported sustained improvement. Now, if we move to bipolar disorder, next slide, we see that 5 of the 5 studies reported sustained improvement when they were followed up over 2 weeks, and they were followed up from weeks to months, actually. Okay. Now, sleep deprivation therapy, possible mechanisms of action, resetting our hypothesis. This hypothesis, resetting abnormal circadian rhythms through clock genes. All right, although the antidepressants, lithium, bright light, sleep phase advance, have many mechanisms of action. One that we hypothesize that may be specifically relevant to sustaining the efficacy of sleep deprivation therapy is that they all have the ability to normalize abnormal circadian rhythms. So deep brain stimulation. This is different from the other 2 that from ketamine and sleep phase advance. And essentially, what this involves is implanting, and I'll show you a slide, implanting electrodes, bilateral electrodes, in specific areas of the brain, in the case that I will focus on most, It's a frontal area called the subgenual cortex, and implanting a sensor or a battery in the chest wall to provide continuous stimulation. Okay, so a little bit about this. The subgenual cingulate cortex is an area called Brodmann's area 25 in the frontal area of the brain, critical area, an area that I've also discussed, predicted hyperactivity of it, predicted the response to sleep deprivation. By deep brain stimulation in the white matter, just adjacent to this structure, can produce rapid behavioral changes in a subgroup of severely depressed patients. While all patients continue to report feelings of moderate depression, this is different contrast to the sleep deprivation and the ketamine that I'll talk about. For several weeks, several patients indicated as soon as they turned on the electricity and stimulated this area, they said, hey, the painful emptiness has gone. The void remitted almost immediately, and that would reverse when they turned it off and then come back when they turned it on. After 1 week of DBS, 40% of the patients in one study responded, and one was in remission. And really the leader in this field is Helen Mayberg. All right, and maybe the hypothesis probably is the acute deactivation of this particular area, which I've shown previously is hyperactive. All right, now this is the standard, the placement of these electrodes is critical. The accurate, and this is the usual 1.5 tester image, much better than we used to have. Some people are now using 3 tester, but this image is a 7 tester. This work has been pioneered by Z.H. Cho, a famous physicist, and you can see a remarkable detail of the brain with these with 7 tester. He actually has just received major funding to develop a 14 tester. I know this is just an animation, and here you see the 7 tester for deep brain stimulation. You see the concept of direct targeting the brain, ultra high resolution, and again you've seen a cartoon of how it might work. Again, prepared by Dr. Z.H. Cho. All right, so deep brain in terms of the antidepressant response, the deep brain can be dramatic, improvement lasting up to a year. However, interesting enough, there are 5 other areas of the brain which have reported response to deep brain stimulation. 27 of 50 responders. And this requires a great deal more investigation, a great deal more work to try to understand it. Helen Mayberg has focused on these 2 areas, and that's where most of the data is at the present time. But there was a follow-up study, nice follow-up study. 20 patients with treatment-resistant depression have received deep brain stimulation to this area and have been followed up for 3 to 6 years. The average response rate at the last follow-up was 64%, and these patients did show marked increased social functioning. All right, now I'm going to turn to the last area before I make a few comments about future studies. Low-dose intravenous ketamine. And this you heard about if you were on the webinar last time. This was discussed by John Kelso. but what? Crystal, I'm sorry, John Crystal. And this was, I'm just going to review some material that he did not cover. All right. So just to set this in a setting here in terms of dose, high levels of ketamine have been used as a psychotomimetic street drug known as Special K. Anesthetic doses in children, and thousands of children have been given this as an anesthetic. Now in depression, and I'll talk about these last 3 or 4, 1 to 2 hour low dose infusions in treatment, 0.5 milligrams per kilogram. And then I'll talk about a very interesting bolus infusion, 1 to 2 minutes in suicidal ideation. All right, so this is just an example of a study by one of the outstanding investigators at the National Institute of Health, Dr. Carlos Cerati. Reported that 71% of 17 patients with treatment-resistant depression, major depression, rapidly improved after ketamine. Patients feel better within hours after receiving an intravenous infusion of ketamine. None of the subjects taking placebo met the response criteria. So this is more invasive than sleep deprivation, but quite tolerable. Now here we have on the left side the 24-item Hamilton scale that I mentioned before, from no symptoms up to severe symptoms. The black line is the placebo, in which all the patients were more severely depressed than this is the black circles, and then the white circles indicate those on ketamine. And you can see this mark here at 110 minutes, the dot here shows significant change, and by 230 minutes, even more change, and that continued. They followed for 7 days. But one of the issues is sustaining this that continues. That I'll talk about. All right. Now, this is possible mechanisms of low-dose ketamine's rapid antidepressant action. And I know this is a technical slide, but it's important, and I'll discuss. Well, it's important because these are clues for the development of other more effective treatment, even more effective. So ketamine, the non-competitive inhibitor of a excitatory neurotransmitter glutamate, and it's a non-effective inhibitor of the receptor that it binds to. Now, other new mechanisms published last year. One is concerning AMPA, again by Dr. Cerati, and it appears to be essential to ketamine's rapid antidepressant effects. The third is Mtor. This was published in Science, and ketamine activates a pathway called the Mtor Pathway. And when the Mtor Pathway is blocked, ketamine behavior response is inhibited in animal models of depression. And then finally, a study of our own. Our research group hypothesized that rapid antidepressant effects of ketamine could involve the modulation of clock genes and circadian rhythms, similar to that we've hypothesized for sleep deprivation therapy. And our new data, published 2 weeks ago, shows that ketamine in fact modulates clock genes in cell culture, and next studies will involve animal models and humans. All right, again, another slide from review by my wife and I, and I'm not going to go through the details, but 15 of 15 studies, some with only one case, report rapid but transient, in many cases, improvement in an estimated 60 to 70% of patients. Now, I want to talk about the studies of use of intravenous low-dose ketamine to treat suicidality. I'll talk about the first and the last study. All right, now just focus on the black line. This is an item. The MADRS is a way of weighting, another scale for rating depression, like the Hamilton. And it has an item on suicidality. And the ratings following a single subanesthetic infusion of ketamine in treatment-resistant mood disorder patients, major disorder patients, with a 24 follow-up. And you can see in the black line here that there was a very significant change in suicidal ideation. This is acute suicidal patients, okay? And you can see that it was sustained. The black line was sustained out to, or this improvement was sustained out to 12 days. The final study that I want to comment on is a real-world study, intravenous ketamine in an emergency room setting at Yale University, which, by the way, Ron Dumas led the paper that I mentioned in ketamine. I mentioned in Science. All right, 12 of 14 responders, 10-day follow-up, lower dose ketamine. Now this is 0.2 milligrams. Ketamine administration as a bolus, first time, over 1 to 2 minutes. Infusion times were different than the usual 2 hours. They did have mild psychotomimetic effects in 2 patients, but they resolved within 40 minutes. Time to significant improvement for the depression was 80 minutes, suicidality 40 minutes, mean time to remission was 240 minutes. Now, this study had some limitations. It was an open study. It used no placebo. Relatively small N. Exclusion, I'm sure the IRB insisted on this, and for this early study, included lifetime diagnosis of psychosis, mania, hypomania. You might be able to get that. Patients on antidepressants of NMDA antagonists. That's just logical. Medications were excluded. All right. So this was their finding. Effectiveness of low-dose ketamine in an emergency room setting for suicidal ideation. And this was open label. 24 patients. Okay. Over here you have, at the time of admission, this is a rating scale. This is the rating scale item for the MADRS, and you can see they're suicidal. And this is the time from 40 minutes up to 6 days in which you have a very significant decrease in the symptoms of suicidal ideation. This was really a bold but real-world study. Very, I think, quite important. Okay, future studies. Identify additional strategies to sustain the effect of ketamine in sleep deprivation therapy. We're working on this. There have been a number of studies, but there's still ways to go in this work. And then high-resolution research tomography, which we have at University of California, Irvine, to identify brain areas associated with rapid antidepressant response. And this is just an example. This is one patient had a dramatic response to sleep deprivation. This is pre-sleep deprivation, post-sleep deprivation, sleep time. And these are MRIs here this is to focus on. But let's just turn the anterior cingulate, which includes BA25 that I've talked about. And you can see in red here that it's markedly decreased after post sleep depth or after sleep depth. So these are just 4 areas that have been hypothesized to be associated with depression and possible response to sleep deprivation and ketamine and deep brain stimulation. This needs to be followed up and future work. All right. So significance. Many clinicians assume that it requires at least 2 weeks and often as much as 10 weeks for significant patient improvement on current antidepressant medications. This field of research could contribute to a major paradigm shift in the treatment of depressed patients, where physicians could expect that 40 to 60% of their patients will improve within 24 hours. Sustained over extended periods of time could potentially reduce suffering and decrease suicidal risk in severely depressed patients. I'm not going to go through these because we're going to run out of time, but these are compounds and strategies, other strategies to test potential antidepressant strategies. I want to note that the NIH has initiated a program, Rapid-Acting Antidepressant Treatments for treatment-resistant patients, to identify pharmacological and non-pharmacological drugs other than sleep deprivation and ketamine, and I've just listed some, in treatment-resistant depression using a network of investigators. So I've reviewed sleep deprivation, possible biomarkers, role of clockines, deep brain stimulation treatment, low-dose intravenous ketamine, future studies. These are collaborators at Irvine. Much of this has been collaborated with UC San Diego, particularly John Kelso here, and you can see the list here. Wu and Potkin, Bunney, Mark Votter, and then sources of funding, Brain and Behavior Research Foundation has been central in starting much of this work. Stanley Foundation, William Lyon Pensner Foundation, Della Martin Foundation. So I've described strategies for treating depression, which have stimulated the search for many new and improved therapies, particularly for severely depressed treatment-resistant patients. And then finally, I would just like to express my special appreciation to the Brain and Behavior Research Foundation for their ongoing efforts to bridge the scientific community with the community at large. So thank you, and we do have a little time for questions. Okay.
Thank you, Dr. Bunney. We really appreciate hearing about these potential new treatments. And one question that I think all of us are kind of curious about, in a depressed bipolar person, can sleep deprivation actually send a person into a manic episode?
Yeah, it's a really good question. And yes, that can happen. We've had it happen, but it's probably as rare or rarer than what you see with the standard antidepressants.
Okay. How is
and that's why in that one study we put the patients on lithium.
Okay. How is sleep deprivation induced, and what are the risk factors in such a
okay, sleep deprivation at the present time requires a nurse assigned to the patient. The patient can read, kind of thing. They can talk. They can, with the nurse, etc. But it does require somebody there. We're trying to work out an outpatient system which would monitor activity. There are a number of new technical devices which monitor sleep and monitor, so we can tell whether the person has been taking naps. Naps really probably are contraindicated during this period of time. So we're working on that. That would be a major breakthrough if we can do that.
And there would really be risk factors associated with self-administration of this type.
Risk factors, virtually none, other than possible precipitation of mania.
Okay. Okay, thank you. We always hear from our constituents with these series that there are pharmaceutical strategies, perhaps, that can be repurposed, go off patent, tweaking it just a bit and selling it as a new product. Is there anything in the horizon related to these therapies that you can think of that you can share?
Well, I probably shouldn't have skipped over that slide so rapidly, but there were about 6 areas that are in development. If you want to go back to that. Okay, that's the slide.
That one, okay.
Oh, so ketamine-like compounds with reduced psychotomimetic effects would be, this has been reviewed by Zarate. 2 people have reviewed AMPA potentiators. And then scopolamine is a possibility, but it takes 3 to 5 days, so it doesn't fit into my, but these are double-blind studies by Drevitz, and, that's a possibility. An interesting one is to give repetitive transcranial magnetic stimulation, which has been approved for depression, but to give it 15 times in 24 hours. One study, but they produced a remarkable response. And then a study, creative study, out of Wisconsin with slow-wave sleep specific blocking it. This was done in patients that weren't very severe, so really needed to be reproduced. But these are, RTMS is available. This takes a lot of technology to do the slow-wave sleep stuff. Scopolamine is available. The AMPA compounds are just being developed, and so are the ketamine-like compounds.
And there's one additional comment that we have. I've condensed several of these. This person says, I just read a headline in the London Daily Mail that stated, why antidepressants are simply a confidence trick, claiming that many mental health professionals believe that taking placebos might work just as well. What are your thoughts on that?
I don't think there's, let's put it this way. There is a significant placebo effect. But if you have a serious psychotic depression, you want to get on an antidepressant. There is no question about it. I would disagree with that. I think that's based on the fact that there have been some studies that show that that only a percentage of patients on their first antidepressant, given for sufficient length of time, that's the caveat, given for sufficient length of time, and at an adequate dose. But there's no question that a significant subgroup of patients respond. I think it was, 40% or so. If you're in that 40%, boy, you want to be on. You want, no matter what percentage you're on, you want to have a adequate trial of 1 or 2 antidepressants. Absolutely.
Dr. Bunney, I think we are about out of time. And I do want to thank you so very much always for your leadership, your work with the Scientific Council, your continuous willingness to participate in events like today's webinar and other activities with the Brain and Behavior Research Foundation has just been so significant over the years. So we are very, very grateful to you for this extraordinary partnership. So thank you.
Let's say these were good questions, too. Enjoyed it.
Wonderful. And, one last thing, for those of you who are listening and are in the New York area, on Wednesday, October 26th, you are certainly invited to our 23rd Annual New York Mental Health Research Symposium. And you will hear, not Dr. Bunney in this particular case, but award-winning, brilliant scientists such as Dr. Bunney will be there, and followed by a Q&A session as well. So you can get on our website to check out information on that. Thank you, as always, for your participation. Again, Dr. Bunney, there are not enough words to thank you for your leadership.
And thank you, Vanita, for being the president and for organizing all these.
And thank you and goodbye.
Bye.